All tables will be disinfected by spraying with 70% alcohol before and after any lab work is performed. Paper towels used for wiping off disinfectant can go into the trash. Hands should be washed before and especially after lab work. Paper towels used for drying your hands can go into the trash. No food or drinks are allowed in the lab.
Ben, you can't do that.
Long hair must be tied back. Shoes must cover the entire foot. No sandals, clogs, etc. No bare shoulders. You can wear a shirt, a sweater, or an apron or a lab coat to cover your shoulders. Eye protection must be worn, goggles, not glasses, and an apron or lab coat. Goggles provided by us are sterilized in the UV cabinet and must be returned to the UV cabinet after every lab. Students may not be allowed to participate in lab without proper attire and protective gear. In your drawer, you can find 6 slide boxes, an inoculating loop, a marker, a clothespin, a ruler, and a checklist of items. Slides or slide boxes may not be taken from the lab under any circumstance. Stain bottles, otherwise known as Copeland jars, are provided in a bin. All staining must be done in a metal tray or the fume hood. Gloves will be provided. Heat-fixed and stained slides are not contaminated. Uncontaminated slides, for disposal, can go to the cardboard box. You may not take prepared slides out of the lab to study at a different location. Wet mount slides have live organisms on them. Do not put these slides in your bag or pocket or take them outside the lab. Wet mount slides must be placed into a dish with disinfectant for disposal. Each student will be assigned a microscope and have a numbered space in the cabinet. Your cabinet number corresponds to the microscope number. You are responsible for cleaning your microscope and reporting any problems with it. Before putting the microscope back into the cabinet, you must wipe off the lenses, set objective to 4x, perform general cleaning of stains on the microscope, and wrap the cord properly. Kim wipes used for cleaning lenses are discarded into the trash. Some things in the lab can be hazardous to you, your group, or the housekeeping staff. These include open gas jets, hot micro-incinerators, chemicals, live cultures, reaching across the bench, not paying attention to your surroundings, 10 to 15 glass tubes in your hands at once, and glass slides in red biohazard bags. Although the bacteria with which we're working with are not considered to be pathogenic in healthy humans, always assume they are pathogenic or infectious and act accordingly. Never ingest or smell bacterial cultures. Cultures provided for lab exercises are found on the side counter. The test tube racks are color-coded for each table. At the end of the lab, collect all the culture tubes and/or plates, place them on the labeled rack, color-coded, and return to the side counter at the end of each lab period. Incubation. If you or your group has more than one Petri plate, stack them on top of each other. Wrap the stack with one piece of tape and print your name or group ID. Place them in the bin for incubation on the side counter. Tubes of media. Label with the tape around the tube just below the cap. Combine tubes in test tube racks, which are color-coded, for incubation. Disposal. All rinsing and decolorizing must be done on the metal tray. Liquid waste from staining/rinsing on your staining tray must be poured into the designated plastic bottle next to the sink for disposal. Do not pour it into the sink. Discarded Petri dishes, after recording results, used swabs, and microfuge tubes go into the red biohazard bags on the tables. Discarded culture tubes, but not broken ones, of media are collected in wire baskets on a cart in the lab. Take the tape off the tubes before placing them on the cart. Used pipettes, the plastic ones, go to round plastic jars on the tables, and the packaging goes into the regular trash can. Immediately report any spills, broken glass, or accidents to your instructor. Any plain broken glass without bacteria, such as glass tubes or slides, may be put into the cardboard box. If you break glass tubes or slides with bacteria, the area must be saturated with disinfectant. Paper towels used for cleaning bacterial spills are contaminated and must go into the red biohazard bag. Broken glass must be swept up or scooped up and put into the metal canister for sterilization. Never put any glass slides or tubes or anything sharp in the red or orange autoclave bags.